All right, so let's go ahead and talk about CRISPR. So CRISPR is very much science in the news, and you've probably seen a bunch of articles recently discussing CRISPR. I'm going to teach you this technology. The more recent article, famously, are gene-edited babies that were edited by a Chinese doctor, and these twins were made to be edited to become resistant to the HIV infection. I believe it was a parent that was HIV positive, and so the twin babies were edited in order to become resistant to this infection. It had to do with the protein that made them more resistant to HIV. So the babies were actually edited while they were in utero, before the mother gave birth to these twins. This basically angered the scientific community because these kind of techniques, these genetically engineering techniques, are quite regulated by the science community, and the scientists in China just went out there and did it, and that was looked down upon. Even though it's cool science, there were many different paths and routes that scientists must take in order to do something that drastic. So if you guys, before I talk about what CRISPR is, if you're interested in CRISPR, you can actually buy your own genetic engineering CRISPR kit. Let me actually show you this on Amazon. So yeah, do-it-yourself bacterial genome engineering CRISPR kit. It comes with these simple tools that you guys can use in order to perform CRISPR technology yourself. And really, this is actually a neat kit that a lot of parents are buying their children in order to have them run these experiments at home. Now, they're not going to edit a baby. Like in the previous example I went over, it just has you edit some things, and it's not going to be as drastic, but it teaches you the technology behind CRISPR and what's really going on with that editing process in a live organism. It's pretty neat. Yeah. Let's go back to our lecture. So what is the CRISPR Cas9 system? The CRISPR Cas9 system is going to be a tool that we use to edit the genome of an organism. I have my pointer here. So here's the DNA. That DNA is going to constitute the genome of an organism. And let's say that there's an area here that I want to edit. It looks like that orange spot right here is a spot that I wish to edit for this organism. Cas9 comes in, and Cas9 is an enzyme highlighted here, that big kind of bean-like looking cartoon. Cas9 is basically molecular scissors, and it's an enzyme that's going to cut a strand in the genome of an organism. It's going to snip it. Where does it know how to cut? The guide RNA that's colored yellow here is going to guide it to a specific spot of the genome in order to cut it. So there's the guide RNA. It is leading that huge Cas9 enzyme to come in here and snip the genome. And that way we have our cut DNA sequence. We can see that they're separated now. And with that DNA sequence now, we can actually disrupt the gene due to that cut. So we delete the gene in the genome of an organism. We can also use that technology to insert a gene quite efficiently into the genome based on that cutting process. So what I'm trying to tell you guys is, with the CRISPR-Cas9 system, this is gene editing in living cells. You don't have to have the editing taking place and then have the offspring show that change. This is a living cell. You can do it in any organism that is alive. This is quite new in the biotech field. Maybe you want to correct a genetic mutation that's harmful for an organism, insert a new gene, turn off an existing gene. So we're talking about GMOs here. This is rapidly becoming one of the most important tools available for genetic engineering. There's a vast array of possibilities using the CRISPR-Cas9 system.